Okay, maybe we should go ahead and get started. Since everybody's here, I'll just say a few words. I know, Anat, you really wanted to have this defense in person. I'm sorry that because of Omicron, we couldn't make it work out, but I'm really looking forward to your coming back in the spring for your graduation and celebrating then. So I'll just say a few words since we are by Zoom. So welcome everyone to Anat's FPO. As you'll see from Anat's talk, she's really done a wonderful machine learning project in the group, trying to predict interaction sites within proteins. But kind of more from like a personal perspective, I wanted to say that Anat brings a lot of organization and careful methodology to the group. And really your thesis work, Anat, is really a model for how careful testing should be done. I'm also grateful for you for bringing many new techniques to the group, including representation learning, which has led to a bunch of different projects in the group. So in addition to Anat's many accomplishments professionally, I wanted to, of course, say that she had 3 big accomplishments while a PhD student, namely she had 3 children. And so it's amazing that you've done all this, Anat. And so with that, let's get started.
Thank you, Mona. Okay, let's get started. So see if my clicker here works. Good morning and good afternoon, everyone. My name is Anat and welcome to my PhD defense. First, I really want to thank each and every one of you for virtually coming to hear my defense during this recent pandemic wave and everything else that's going on today. As Mona said, I no longer live nearby and was planning to fly to Princeton and defend in person. But thanks to Omicron, I unfortunately had to cancel my flight and use the great invention called Zoom to complete this very crucial part of my PhD. So these are crazy times indeed, but some might call us crazy for doing a PhD at all. In my defense, as the name alludes to, I'm going to talk with you and show you how I use machine learning and data aggregation techniques to predict protein interaction sites. So since this talk should represent my dissertation, I give you here another way of summarizing my dissertation. So this is a brief summary of what this talk is going to be about. I will talk a lot about proteins, domains, their binding positions, and how we used sequence features to make predictions for different ligands. Now, instead of looking at a cloud of words, let's start from the beginning. So in today's talk, I will first give some background and motivation for the overarching problem we're trying to solve, our general approach for solving this problem, and how our work fit within the realm of other efforts in the field. Then the bulk of the talk will be discussing the 3 components of my thesis, which are 1, building a domain-based aggregation framework to differentiate binding positions. 2, constructing a supervised machine learning framework for prediction of domains interaction sites. And 3, building models based on pre-trained protein deep models for predicting interaction sites in proteins. And lastly, I will discuss my conclusions and some ideas for future work. Okay, let's start with the introduction. Proteins are the product of the genetic information stored in each of our cells. According to the central dogma, the instructions in our DNA are converted into a functional product called a protein. Proteins facilitate nearly every task of cellular life, including catalyzing chemical reactions, giving cells their shape, and performing routine cell maintenance. They also receive signals from outside the cell and mobilize intracellular responses by binding to a variety of binding partners referred to as ligands. I'm only showing here a few examples, as the scale and diversity of proteins' functions are astounding. Remarkably, all the proteins are comprised of a simple set of 20 amino acids, and their order is determined by our DNA. The majority of proteins do not work in isolation. They function by interacting with different molecules in our cells. Some proteins form complexes with other proteins via stable interactions, taking part in permanent complexes as subunits. Other proteins interact in transient interactions to carry out functional roles such as signal transduction. Many proteins bind ions and other small molecules to catalyze vital metabolic reactions or to stabilize a structure required to perform a certain function. Some proteins, including transcription factors, bind highly specific DNA or RNA subsequences to accurately determine gene expression regulation. Bless you. Over the years, many experimental methods have been developed to investigate protein interactions in vivo and in vitro. These experimental techniques, while vastly expanded our knowledge of cellular functioning, have many limitations. They're usually expensive and complicated. They take a lot of time. They are also limited to certain types of proteins and interactions we can manipulate and test in the lab. And moreover, they're usually not designed to identify the specific protein positions that mediate the interaction. Identifying the specific positions that mediate an interaction is critical for understanding of protein molecular mechanisms. For example, this is an illustration of the zinc finger, a well-studied domain found in proteins that bind our DNA and regulate our gene expression. It binds a zinc ion using 2 histidines and 2 cysteines to stabilize its structure. Identifying the exact binding positions is also critical for understanding the impact of changes in the amino acid identity in one of these positions. Mutation in one of these positions, demonstrated by these yellow stars, can destabilize the protein structure, lower its propensity towards its ligand, or even cause the protein to prefer a different ligand. At the same time, a change in other positions may have little to no effect on the protein's function. Each of the 20 amino acids has different characteristics that allow this part of the protein to bind other molecules, or allow the protein to fold in a certain way, or to move through a certain medium in our body. For example, because the DNA is negatively charged, the amino acids involved in binding DNA are usually positively charged amino acids. You can see here in this image that amino acids can be grouped according to their physiochemical properties. And we'll talk about this again when we think about features that differentiate binding positions. Identifying the specific residues is important, however challenging, since these positions may not be proximal with respect to the linear protein sequence and nearby only when the protein folds to its 3-dimensional structure. Therefore, the most accurate way to identify these interaction sites is through analysis of experimentally solved protein ligand co-complex structures. Solving them requires laborious, time-consuming techniques, and therefore structures are not available for the majority of proteins to date. For example, if we look on the human genome, well, we have roughly 22,000 genes and relatively high coverage to other organisms. Only 13% of them are associated with biologically relevant protein ligand co-complex structure. We can double this coverage using homology modeling. And with these inferred positions, we still only cover 27% of the whole genome. Interestingly, if you look at proteins that contain a domain with a relevant co-complex structure, then we can cover almost 2/3 of the human genome. And I'll thoroughly explain about protein domains in the next slides and how we use protein domains in our framework. But the point here is that we still do not have enough protein ligand co-complex structure to cover all our protein sequences. This is a good place to mention the AlphaFold 2 algorithm developed by DeepMind that can predict protein structures highly accurately. One would hope that this algorithm would simply close the gap over here. However, their algorithm is not focused on protein ligand co-complex structures, and without a ligand, it's a little trickier to identify interaction interfaces. In contrast, we live in the era of genomics and big data. The decreasing price of DNA sequencing has resulted in accumulation of an enormous amount of sequencing data. The pace of sequencing far exceeds our ability to characterize each of these proteins experimentally. To narrow this gap to functionally annotate all sequences, computational methods play a key role. We wish to exploit this abundance of sequencing data, but also utilize the valuable information obtained from solved structures, and in particular, the pair residue resolution obtained from structures. So our approach is building machine learning frameworks that can use structural labels and integrate it to newly predict interaction sites from sequence alone. Before I go into details, I briefly summarize here my thesis contributions. First, I relied on scientific knowledge accumulated about interaction sites properties and performed a rigorous curation and analysis of unprecedented scale by constructing numerous sequence-based features that differentiate interaction sites. These features are derived from different data sources in different aggregation levels. Next, I used these features and a domain-based aggregation technique and built a comprehensive machine learning framework that relies on structural labels to predict interaction sites in protein domains. Our framework demonstrates high precision, and we newly predict interaction sites for 467 domains with unknown function. Finally, I explored the potential of building featureless models from pre-trained deep models to predict interaction sites in proteins. Our models demonstrate high precision in pair residue and whole protein predictions. Different parts of my thesis were also presented in the following journal and conferences. In particular, the work presented in the first two chapters was published last year in NAR. Okay, now I'll move on to describing what protein domains are and how we use them to amplify signals that differentiate binding positions. Protein domains are subcomponents of proteins that are grouped into families that share sequence, structure, and evolutionary descent. More often than not, proteins accomplish their function through one of their domains. This drawing of black lines depicts a protein sequence, and a protein can have multiple domains, sometimes of the same type and sometimes of different types. Here we can see several real protein domains with their structures colored white or partially red, and a ligand that they bind colored in blue. So for example, DNA, RNA, ion, peptide, and small molecules such as ATP. Another interesting thing about protein domains is that they can be identified from sequence alone. In fact, they are presented as hidden Markov models. And this is again an example of the zinc finger domain, this well-studied domain that binds DNA and the zinc ion. We have here several aligned sequences of the zinc finger domain from different proteins, from human and other species. And we can see that some of the positions are conserved. Then, if we look on the sequence logo representation generated from the H&N, we can see that where the height of the letter is high, it means high conservation. And we have 4 such positions, those 2 C's and 2 H's. And if we look here on the structure, these are the 4 positions in contact with the zinc ion here in green. So like I already showed you in the introduction. And I already mentioned that the zinc finger binds DNA, but it's not so clear from this logo where these positions that bind the DNA might be. So these are actually these 4 positions. And we can't identify them based on sequence conservation. So this is why this task is not so trivial. We have decided we will try to use machine learning algorithms for identifying these positions. But our first task was gathering a comprehensive set of features that will enable these algorithms to perform well. We started by data analysis and curation across the human proteome. For each protein domain, and I use here as an example our favorite domain, the zinc finger, we can find its HMM profile in PFAM, a standardly used database for protein domains, and use Hammer to identify it across all the human proteome. Then we aggregate all the instances we find, and for each HMM match state, we compute a large set of features in 3 different aggregation levels. Some of our features were based on the corresponding DNA sequences. For example, the variation across healthy human population or the conservation across different species. Other features we derived from the protein sequence. For example, the distribution of amino acid, predicted structural features from using external tools, and also protein level conservation. Many features are also encoding physiochemical properties of the amino acids. Remember I said earlier that different amino acids can have different charge, for example. The charge can affect their ability to bind certain molecules. And so it is a property that can be useful for machine learning algorithms to differentiate these positions. The third aggregation level is the domain. And we can also encode features directly from the HMM profile. For example, the state's emission probability for each of the amino acids. Then our features are aggregated according to the level in which we acquired the features. So DNA level features are aggregated to protein positions, and together with the protein level features are aggregated to domain positions to create a table of features calculated for each of the domain's positions. So in the illustration here, the zinc finger domain has 23 HMM match states. So each of them will be a sample that we encode features for. Now, in addition to features, another important component of supervised learning is the ground truth labels, which, as I mentioned earlier, are coming from protein ligand co-complex structures. Here are the same domain structures with their ligands I showed earlier. Only now you can see that there is a meaning to the color. It represents the fraction of structures in a protein ligand co-complex structures database called BioLip, where the position was in contact with the ligand. So, for example, a score of 10% represents that in 10% of the structures of proteins containing this domain, this position was contacting the ligand. And the higher the score, we are more likely to believe that this position is mediating the interaction. If you look here closely, you can see that the red parts indicating higher score are the ones touching the blue ligand in each of these cases. This work of systematic aggregation and scoring of domains positions by Coburn and Singh was recently published from our lab, and I'm using these scores as my labels for the protein, for the domain's binding positions. We constructed features in generally 6 functional groups. Number of features is listed in orange. We encoded conservation in all levels, DNA, protein, and domains. Physiochemical properties of different types, including hydrophobicity, charge, and hydrogen bond potential. Population variation features were based on many statistics we did on the type of variation and their frequency across healthy human population. We used external methods for structural prediction and encoded features including surface accessibility and backbone angles. Location-based features depicted information on where the position is located within the domain and where the domain is located within the protein. And finally, we also newly constructed several information theoretic measurements that can quantify selective pressure based on the population SNPs we observed. And also not included in the numbers here are additional windowed features that we added for some of these features. We labeled all the positions within our domains as binding, non-binding for 5 different ligand types, and then assessed how well each of them differentiate between binding and non-binding positions using a Wilcoxon test. I can't really go over hundreds of features, but I'll give a few examples. So here's a feature representing the hydrophobicity of the domain positions, where for each ligand, we examine the difference between the distribution of binding and non-binding positions. And the p-values presented are Benjamini, Hochberg, FDR corrected. We can see that binding positions, which are in the darker colors, are significantly less hydrophobic than other positions. This can be attributed to the fact that hydrophobic positions are usually buried in the core of the protein, whereas interactions usually form on the surface of the protein. Another example is looking on the polyphen score. That's a score that represents the possible impact of an amino acid substitution given to each SNP occurring in human healthy population. We can see that the number of SNPs with a probably damaging classification tend to be significantly higher for binding positions. Because as previous studies show, disrupting a binding interface usually damages the function of the protein. And here's an example of mean surface accessibility obtained from predicted structures. We can see that nucleic acid binding positions are significantly more exposed on the protein's surface. And in contrast, the ones binding ions and small molecules tend to be less exposed. And this can be attributed to the fact that proteins usually bind these small ligands via binding pockets. Altogether, the majority of our 753 features significantly differentiate binding positions across all 5 ligand types. And this is just the effect of each feature individually. What if we combine them? Now I'll explain how we utilize this great resource of sequence binding features to develop a machine learning framework for predicting binding positions. Our machine learning framework gets us an input, a protein domain HMM sequence. And as mentioned before, we constructed the following features table where each domain position is assembled, for which we have features and a ligand type binding labels. Now, all our big table of features is fed into this ensemble of classifiers, which I will talk about next. And in the end, we receive a binding score for each of 5 ligand types: RNA, DNA, ion, peptide, and small molecules. We called our framework DSPRINT, stands for Domain Sequence-Based Prediction of Interaction Sites. Now I'm getting to the core of the machine learning framework that I built to predict binding scores for each domain position. So first, to ensure I'm not overfitting my data and still able to evaluate my performance, I'm doing the very standard procedure of splitting my data into folds. In my case, 5 folds. I'm training on 4, predicting on the test set. This is assuming I already found my hyperparameters. This is done for each one of 5 different ligands. Each ligand has a different target variable that I'm training on. So these are 5 different classifiers. But for each ligand, I also use 5 different machine learning models: XGBoost, logistic regression, support vector machine, random forest, and neural networks. So if you think about it, these are 25 different classifiers, and each of them return different binding scores as a result. Now, I've decided to add a second layer of prediction using a meta classifier in an architecture called stacking. This is a common practice in many models and data science competitions, such as Kaggle. And what it means is that we take the outputs from the first level classifiers and we use them as additional features. This is usually done when using different machine learning algorithms. Each of them might have different strengths. For example, random forest, which is a nonlinear model, might capture things logistic regression doesn't, and vice versa. What is interesting in our case is that we also have different target variables that we are combining. Each ligand has different labels that we're trying to predict. But it could be that knowing what is the score for binding ion can help in predicting the binding of DNA. Usually, in most prediction tasks, we're only optimizing one goal. And here I have 5 sets of labels I'm trying to learn, one for each ligand. But there are also deeper connections between them biologically. So we are using information from the other ligands to improve our predictions. You can see here that if we look on all our 25 models as a square of models versus labels, the color and pattern represent which ligand and model were used, and each of the 25 models have a different output. We can decide which of them we're combining together. It doesn't have to be all of them. I won't get into the different architectures we tried here, but you can see, for example, that I can choose to use just one model and all the ligands, or just one ligand and all the models. And after we have decided on the stacking architecture, we add these outputs as additional features and take the meta classifier and train on the improved features table. We can keep adding more and more stacking levels in the same way, but since this is not a data science competition and we're not interested in squeezing every little bit of performance, there is also a trade-off here between increasing performance on the account of interpretability. Here, I'm showing the results of training all my models on all my ligands, which are RNA, DNA, ion, peptide, and small molecule, in 5-fold cross-validation, including the tuning. I'm showing here the results of for 5 regular base models and 4 stacking architectures. What is calculated on the y-axis is the AUPRC, which is the area under the precision recall curve, divided by its baseline. And the baseline here corresponds to the fraction of positives for that ligand. So basically, within each subplot, the bars are comparable. The name convention for the stacked models doesn't matter here. What I want to focus on is that the stacking usually outperform the regular models, sometimes substantially. And we can see here that the stacked models achieve significant fold improvement over the random baseline. And this is a different way of looking on the performance. Since the standard use case of our framework is predicting for individual domains, we're interested in how our performance looks like within each domain individually. So in this plot, every domain ligand pair is a dot colored by the ligand type. And for each domain, we calculate both AUC, which is the y-axis, and the AUPRC fold improvement, which is the x-axis. You can see here that the majority of the domains are above AUC of 0.5 and above AUPRC fold improvement of 1, which are sort of baselines for random performance. In the table below, I list the exact percentiles for this case, and also the percentiles for domains achieving a good performance of AUC above 0.85 and AUPRC fold improvement above 1.5. In the figure, I also labeled 3 domains for each ligand type that were above AUC of 0.85. And you can see here that many of them are at AUC of 1, which means perfect ranking of binding and non-binding positions within the domain. Now, when comparing this print to other methods, we couldn't simply get the other protein domain-based predictors and run them on our data because we haven't found any tool designed specifically for domains binding positions. Instead, we used protein-based predictors and aggregated their predictions for each domain position, then took the mean. I'm showing here ROC and PR curves for predicting DNA and peptide binding positions in comparison to 2 other relevant methods. We can see clearly in these ROC and PR curves that this print, colored here in red, outperformed the protein-based predictors. I also want to note that aggregating the predictions of the other methods per domain position and taking the mean is just one possible way to use their predictions for protein domains. and it might not be the optimal way. However, taking the predictions means it is quite a reasonable way to make this comparative analysis. Now I'll discuss some of the cool things we discovered using our predictions. Many proteins domain do not have any functional annotation. In fact, PFAM, the domains database, labeled these positions as DAF, domain of unknown function. We ran this sprint on all 467 DAFs that are not having structurally inferred labels and found several potential binding domains for each ligand type. To give a few examples, this sprint identified several highly scored RNA binding positions in DAF 1754. You can see here a screenshot from our dsprint website, which is publicly available for anyone who wants to query a protein domain and get our prediction scores for all 5 ligand types. So you can see here in red, the RNA binding scores predicted by dsprint. Dsprint didn't predict any other ligand to be bound by this domain. Now we found that DAF 1754 is also the only domain in protein FAM32A, a known RNA binding protein, since it is a subunit of the human spliceosome. Also, interestingly, in the recent PFAM version, this domain was renamed to FAM32A. And another interesting example is DAF 1220 that has recently also changed its name to the Ulduvai domain. Here our website already reflects this name change. And we can see here that these sprints predict mostly DNA binding functionality, colored here in blue, also a little peptide binding, colored here in purple. This domain is found in NBPF proteins and is notable for its lineage-specific expansions in human, its correlation with brain size, and its putative association with neurological disorders such as autism. And while the functions of this domain and the NBPF proteins are unknown, this sprint predicts that the DAF 1220 binds DNA, which is consistent with the intriguing proposal that the NBPF protein could be a transcription factor. Now, these are of course just a few examples we could corroborate from the literature, while other predictions might represent completely unknown interactions and can be an additional line of evidence driving an experiment that will validate these interactions. Now we're going to switch gears and I'll talk about a different angle to predicting interaction sites in protein sequences using pre-trained models and no supervised training features. Natural language processing, abbreviated as NLP, is a subfield at the intersection of linguistics, computer science, and machine learning that aims at creating algorithms that can process textual natural language data and understand the contents of documents and the different nuances of the language within them. In the last couple of years, the NLP application space has seen a burst of novel deep learning architectures, driving sustained improvements in state-of-the-art learning capabilities. This trend was mainly driven by the discovery of attention-based deep learning architectures, which are implemented by transformers. The principal idea behind these approaches is a process called pre-training, in which a model is trained extensively on a very large number of sequences. So in the case of standard NLP tasks, this large corpus could be, for example, Wikipedia. In the case of protein research, this large corpus could be a large number of protein sequences, for example, UniProt. The pre-training process is self-supervising, meaning this training does not use external labels, but instead the models are trying to predict hidden tokens within the input sequences. Then, these pre-trained models can be used in a supervised learning process called fine-tuning, in which the pre-trained model is trained with a labeled dataset for a specific downstream task, In the case of pre-trained protein model, this data set can be, for example, protein sequences with labeled interaction sites. Other examples might be labeling the sequences with secondary structure or subcellular localization and even Go annotations. Then the fine-tuned model can be used to make predictions for the downstream task of interest. These pre-training efforts are usually computationally intensive and time-consuming, but in an effort to democratize AI, many groups have made their pre-trained models publicly available to speed up protein research. Also, several studies have shown that molecular and functional properties can be extracted from the pre-trained models even without any further supervised training. That led us to believe that we can utilize these protein pre-trained models for our goal of predicting protein interaction sites. Remember that one of the obstacles in our training, and generally in many biological data sets, is the abundance of sequences on the one hand, and on the other hand, the limited number of labeled sequences due to the high cost and challenges in acquiring supervised protein labels. This approach enables the model to benefit from the increase in protein sequences, which are used in the pre-training step, and get a more representative glance on the proteome than standard models that are trained using just the labeled sequences. Another way to look at it is taking a neural network that instead of initializing all its weights at random, the weights are initialized in a more optimal way after an extensive observation of an entire corpus of sequences. In my dissertation, I focused on the second step of doing fine-tuning to predict protein interaction sites. We explore this approach using BERT pre-trained models. BERT uses the self-attention mechanism. Briefly, during the pre-training, BERT masks parts of the input sequences and tries to predict them. BERT also became the de facto standard for transfer learning in NLP and is fine-tuned for a variety of NLP benchmarks. More specifically, we're using the prot-BERT BFD pre-trained model that was recently published from the Ross lab. This pre-trained model is based on BERT and is pre-trained on the big fantastic database, which after similarity filtering and clustering resulted in 2.1 billion proteins and 393 billion amino acids. The general architecture of the prot-BERT model is outlined in this figure. Briefly, BERT was pre-trained with a BFD dataset with a higher number of layers than the original BERT publication. And the authors then fine-tuned their model for a few downstream prediction tasks. One of them is, for example, pair residue prediction of secondary structure. To do that, the outputs of the last attention layer of their model were used to derive a 1024 size dimensional embedding for each protein residue. And then the embeddings were input into a 2-layer convolutional neural network to make their pair residue predictions. Now, we are using the pre-trained model in 2 different approaches. The first is fine-tuning the pre-trained model in the standard way I presented earlier. We take the pre-trained model, train it with our labeled data set of protein sequences with interaction sites labels, and the output is the final model we can now use to predict protein interaction sites. The second approach is we're only taking the embeddings from the pre-trained model last layer. Then we use them as the input to a customized network, which I'll get to in a sec. And this architecture is the one we train to make the final model. The architecture we built gets us an input, the protein embeddings extracted, 1 embedding vector for each protein residue. These inputs are fed into a bidirectional LSTM. LSTM is a type of recurrent neural network that is highly effective in addressing the vanishing gradient problems typically encountered in traditional recurrent neural networks. And the bidirectional LSTM is essentially two LSTMs in opposite directions, one taking the input in a forward direction and the other in a backward direction. And this basically allows information from both sides of the protein residue to influence the prediction, similarly to how residues on both sides of the protein chain can affect its binding properties. We then feed the output into two fully connected layers, and then we predict an output of 5 binding scores, one for each ligand type, for each protein residue. An important thing to note here is that our customer's architecture is multi-label, so it predicts simultaneously for all the 5 ligand types. And here we present the performance of all our models on the independent test set, after we carefully split our data to training, validation, and test datasets and tuned hyperparameters. The results reported here are the AUC, AUPRC, and AUPRC fold improvement for each of the ligand types. Now we can see here that both transfer learning approaches are achieving high AUPRCs and AUCs, particularly for the nucleic acid binding predictions, but also for the other 3 ligands. And we consider the fine-tuning model, which is our first transfer learning approach, as performing slightly better, since for 4 out of 5 ligands, the fine-tuned model is achieving higher AUPRC. AUPRCs are primary metrics, and for imbalanced datasets, AUC tend to overestimate the performance. Now, overall, it seems that despite not using any feature engineering and feeding the model the protein sequence alone, the models are able to fit our data and make reliable predictions. We also used our models to predict whether the protein in general binds a specific ligand type. For our test dataset, consisting of 1,926 proteins, we labeled as protein binding all the proteins with at least one residue binding that ligands. So these are the true binding proteins shown here in a red circle. And assessed the proteins that had at least one residue predicted to bind that ligand. And these are the predicted binding proteins shown here in a blue circle. And the overlap between our predictions and the true labels is listed with a line. This overlap consists more than half of our predictions across all ligand types. And so our predictions have a high precision. Our models predict a slightly smaller number of proteins to be binding. And for RNA, DNA, and peptide, our predictions are in the same order of the number of true binding proteins. So overall, our predictions are very reliable and demonstrate high precision also for whole proteins. In general, this idea of self-supervision holds great potential, and I believe it can advance protein computational research in other applications where acquiring labeled sequences is still a great challenge. Now, after telling you about all the different approaches I used in my dissertation to bridge the gap of functional annotation and protein interaction sites prediction, I'll summarize my findings in a few words. To conclude, in my dissertation, I've introduced several approaches for predicting protein interaction sites. These approaches vary in several aspects. The first is data aggregation. My methods leverage protein sequences and also protein sequences aggregated per domain family. The second is the choice of prior knowledge our models rely on, which can be carefully crafted features or featureless models that rely on the sequence alone. The third is the computational technique used to identify interaction sites, whether it is statistical information theory measurements that highlight binding positions, a supervised model that uses a labeled dataset to predict interaction sites, or transfer learning from self-supervised model that is used for downstream interaction sites predictions. Through these approaches, I've expanded our knowledge of protein interaction sites And I also methodically explored many approaches to obtain better predictive power in predicting protein interaction sites. Each of these approaches serve as a stepping stone towards improved methods for interaction sites prediction. The binding features we constructed can be used, for example, in statistical frameworks for rapidly prioritizing positions with binding potential. They can also be incorporated as features in different machine learning algorithms. The disprint framework can be extended to a larger training set that includes additional organisms, as it currently focuses just on the human proteome. Now, given a substantially larger data set, neural networks may improve upon the stacking-based architecture, since their performance tends to increase with data size more rapidly than traditional machine learning algorithms. The predictors we built upon the pre-trained models can incorporate more labeled protein sequences during their fine-tuning steps. Also, the multi-label architecture might benefit from increasing the number of embedding layers obtained from the pre-trained models. And other deep learning architectures might improve our prediction performance as well. Now, our predicted interaction sites can advance research in several avenues. They can help prioritize disease-causing mutations, since many diseases are driven by mutations in protein interaction sites. A better understanding of disease mechanisms can also advance drug design, as drugs typically target protein binding interfaces. Our methods can also be applied to study complex microbial communities by identifying the small molecule mediated interactions that govern these systems. Also, metagenomics may benefit from our domain-centered approach by functionally annotating HMM profiles built from uncharacterized gene families. In conclusion, this dissertation deepens our understanding of protein molecular interactions by developing novel methods for predicting interaction sites. Additionally, this thesis is an important step towards advancing the ultimate goal of functionally annotating the entire proteome. And with that, I'd like to end my presentation by acknowledging all of Mona Singh's lab members, past and present during my time here, and also my other collaborators and sources of funding. I especially want to acknowledge David Todd, a Princeton undergrad whom I first mentored through the rematch program, and we later continued to work together for another 3 years, and is also the co-author of my NAR paper. Now, before we jump into questions, this presentation, other than presenting nearly 6.5 years of hard work, also marks the end of a chapter for me. And I want to use this opportunity to acknowledge the people who have helped me along the way. So first, I want to express my deepest gratitude to my advisor, Professor Mona Singh. Mona was highly involved in all the small details of all my projects, and she mastered to perfection the balance between being very involved and hands-on, but also giving me my academic freedom when the time is right. During the years, Mona has also always supported me with the challenge of raising young kids while in grad school. And it is through her help and support that I managed to thrive on both aspects. I learned from Mona how to do a solid and sound research, and she has helped tremendously in shaping me into the researcher and scientist I am today. And I also want to thank my lab mates who shared with me long hours of research, frustration, and small victories. Already from rotating in the lab, it started to feel like home. And I officially joined the lab at the end of 2015. Now, during my first summer, I started this routine of summer lab barbecues, a tradition that will hopefully live long after I'm gone. And you can see here that we made sure to meet and eat every year. Well, almost every year except the infamous 2020. I miss you all so much. And I wish I could be with you all in Princeton during this huge milestone. Our lab is an amazing group of highly talented people, and you all have gave me so much strength and inspiration that drove my thesis to completion. And I want to thank my friends from the US and from Israel. You know who you are. Many of you gave me meaningful advice on various of personal and professional topics, and this has helped me tremendously during my years in grad school. And I also thank my parents and my 3 sisters who were always there for me, believing in me, even though the distance was tough. And I also thank my more extended family, my in-laws, and especially my cute nephews, who I finally enjoy seeing them grow up in the flesh now that I moved to Israel. And lastly, no words can describe my gratitude to my husband, Adi. We arrived at Princeton together, started our PhD side by side while raising 2 cute then babies, our twin daughters, Noga and Ella. In this photo, you can see Adi in his FPO, which was a little different than mine in the old-fashioned pre-Zoom way. Noga and Ella grew up having a PhD student mom who they didn't get to see every day during stressful times. And I hope that one day they will be proud of me. And lastly, there's Gaia, our newest sunshine in the house that made the year 2020 so much better and happier for me. I love you all. And thank you all for your time.